So today we're going to look at DNA replication. At first, we're going to talk about the purpose of DNA replication. We all know that cells need to make a copy of DNA before dividing, so each other cell has a complete copy of genetic information. And in this process, the parent cells need to make sure the accuracy of the genetic information. So the scientists have proposed 3 different replication models to explain this process. They are semi-conservative model, conservative model, and dispersive model. All 3 methods of replication are interesting, but before looking at these models, we have to know the structure of chromosome. Look, this is a chromosome, and they are telomeres. Telomeres are functionless but repetitive sequences of DNA at the end of chromosome that protect genes from the chromosome shortening that happens at the cell division. So these telomeres are functionless, but they are very useful. These are genes for different characteristics. They are very useful, so they are functional genes. And this is a centromere. Centromere holds the 2 chromatids together, so we know that a chromosome consists of 2 chromatids. And next part, we are going to look at Malthusson and Stahl experiment. These are 2 scientists who set up a particularly ingenious experiment to test how DNA really replicates. At first, we're going to talk about these 3 models. At conservative replication, the DNA parent chain is opened by the DNA helicase, and the daughter chain is copied according to the contents of the parent chain. As we can see in the picture, after the first round of replication, 2 daughter cells are formed, one containing the complete parent strand and one containing all newly synthesized DNA, and so on. After the second round of replication, 4 daughter cells are formed, one containing the parent DNA, and other 3 are all newly synthesized DNA. The second method is semi-conservative replication, which is now the most recognized model of replication. The parent strand DNA is separated, and each parent strand is then paired with a newly synthesized DNA strand, like this. This method can effectively ensure the stability of the daughter cell genetic material. After the second round of replication, 2 of the 4 cells we got had a silver and parent strand each, and the other 2 were made of newly synthesized DNA daughter strands. The last and most complex model is dispersive replication. The parent strand of DNA is broken down into many tiny parts, like this, which are then paired with the corresponding daughter strands of DNA. So the cells that replicate in the first and second rounds both have part parent chain and part daughter chain. To test how DNA really replicates, the 2 scientists set up a particularly ingenious experiment, like this. They first cultured the bacteria in the solution containing nitrogen-15, which is a heavy nitrogen. So each bacterium carries nitrogen-15 on both strands of its DNA. Next, they put the bacteria in the solution containing light nitrogen and let the bacteria replicate 1 round. After 1 round of replication, they take the bacteria and look at the ratio of nitrogen-15 to nitrogen-14. After the first round of replication, they observed that the bacteria's DNA was medium weight, like this one, which ruled out the possibility of conservative replication. Because in the model of conservative replication, you should end up with 1 light and 1 heavy DNA, 1 light and 1 heavy DNA, not 2 medium DNA. Then they performed a second round of culture and found 2 medium and 2 light strands of DNA, like this figure. At this point, we ruled out the possibility of dispersive replication, because if we follow the principle of dispersive replication, we should get 4 strips of DNA that all fall somewhere between light and medium wave like this place. So now we know the accuracy of semi-conservative model.